Hello guys, welcome to Dr. Science. So today I am going to talk about what is meant by glycolysis. So before going into the topic, I would like to tell that you should know what are the different types of blood transporters. I'll be making a separate video on that one because it is a very long topic. So you should know the different blood transporters. And in this video I'll be mainly talking about what are the enzymes involved here and how many ATP and NADH are formed here. So first you should know, I know this diagram here is very complex, but I will break it down for you. So first you should know that what happens when you take food. When you take food, you are taking directly glucose, right? You are taking the glucose from the food, or you are taking fructose from the food, or you are taking galactose from the food. I will not talk about these, but glucose is the main thing we want here in the glycolysis. So what is happening to this glucose which is taken from the food? So this glucose enters mainly three cycles. The first one is glycolysis, and from this glycolysis it will enter into Krebs cycle, and from this Krebs cycle it will enter into electron transport chain, and finally in the electron transport chain, oxygen, and from the oxygen we breathe oxygen, right? Oxygen is a electron acceptor, so it will accept the electrons, and finally ADP is converted into ATP. So what is happening? The glucose is giving ATP by these three cycles, which is glycolysis, Krebs cycle, and electron transport chain, and finally we are getting energy from the electron transport chain. So what is the function here of the glucose? So glucose is indirectly giving us energy, which is in the form of ATP in the electron transport chain. I'll be making a separate video on the Krebs cycle and the electron transport chain, but please remember that glucose not only goes to the glycolysis pathway, it has to go to these three pathways in order to gain ATP. So first we will see what is happening in the body. Okay, so suppose this is your stomach, and you have taken food in the morning. You have taken like 2 pizzas, okay, and pizzas are containing some amount of glucose. So what is happening? This glucose is getting absorbed. So here are the liver. This liver has certain types of receptors or transporters. We call them as glucose transporters, okay, or glut. Okay, glut means the GLU stands for glucose and T stands for transporters. So these glucose transporters, what they will do? They will accept this glucose, and this glucose is taken into the cells of the liver. Okay, so what is happening in the liver we will see. First you should know the structure of glucose. Glucose is as you will be hearing in the school days itself, it is C6H12O6. I will not draw the entire diagram for this one. So C6 means it is containing 6 carbons, 1 2 3 4 5 6. So suppose if this is the structure of glucose, which is containing 6 carbons. Suppose there is an enzyme which is keeping a phosphate on the 6th position. So what it is called? Glucose 6 phosphate, right? So that is how you remember these things. Now we will take what is happening in the cell. So here is a blood transporter, and here is glucose, and once it enters the cell, here is the 6 carbon compound which is glucose. Okay, so what happens once it enters the cells? So once it enters the cells, we have to add a phosphate. Why are we adding phosphate? I will just explain to you. But take a look at the diagram here. 1 2 3 4 5 6. So on the sixth position we are adding a phosphate. Why are we doing this phosphate? Can you guess it? We are adding the phosphate because phosphates are negatively charged, and by adding this phosphate on the glucose, we are letting the glucose to stay inside the cell. Suppose this is the cell. We are making sure that the glucose won't escape this cell. Okay, by making a charge, by adding a phosphate which is negatively charged. Okay, so that's why we are adding a phosphate here. Okay, so what is the enzyme here? The enzyme here is hexokinase or glucokinase. Hexokinase is the enzyme which is mainly present in the muscles. Okay, I'll be discussing about hexokinase and glucokinase in the blood transporters, because you will be hearing the words about high VM and high Km. Okay, so you should know what is meant by that. I'll be discussing that one in the blood transporters. So glucokinase is the enzyme which is mainly present in the liver. So what is the function of these kinases? So what are meant by kinases? Kinases are nothing but, suppose imagine this is a guy. The function of this guy is to put one phosphate. Okay, suppose he is holding with this hand a phosphate, and this is a car. Here is a car. Suppose this is a bike. Okay, so the function of kinases is nothing but they take this phosphate here. Okay, they remove the phosphate from the car and add this phosphate on the bike. That is the function of these kinases. Kinases are nothing but they do, we can simply say that they do phosphorylations. Okay, so what is the function of kinases? They simply cut this phosphate from one substrate and add this phosphate to another substrate. We will take an example here. I already told you glucose is a 6 carbon compound, right? So what is this glucokinase or hexokinase doing? There is ATP. What does ATP stands for? ATP stands for adenosine with 3 phosphates, right? So what this kinase will do? He's a bad guy. He will cut this phosphate. So ATP. So once ATP loses its phosphate, one of the phosphate, it is called as adenosine diphosphate. Since only 2 phosphates are present, ATP is converted into ADP. Okay, so this kinase guy, he is breaking this phosphate and he is converting it into ADP. So what will happen to the phosphate here? That phosphate is getting added to the 6th position of the glucose. So it is getting phosphorylated at 6th position. We call this glucose as glucose 6 phosphate. Okay, so that is the function of these kinases. So whenever I say kinases, they are taking a phosphate and doing phosphorylation to another substrate. Okay, like next, what is the glucose is the first reaction here is glucose is getting converted into glucose 6 phosphate, and this step here, this first step is very important because it is very highly regulated. Okay, It means the insulin, you will be hearing insulin and glucagon. They will be controlling mainly these steps in the glycolysis pathway. So first step in the glycolysis is highly regulated. You should know this one. You will be hearing this one in USMLE. So what will happen? Glucose 6 phosphate. So here is a glucose molecule, and there is a phosphate attached here. So glucose 6 phosphate is converted into another compound called as fructose 6 phosphate. Actually there are small hydroxylation, but this step is nobody asks about these steps. Normally glucose is converted into fructose in this step by the enzyme glucose phosphoisomerase. Isomerase is the enzyme which will do some kind of hydroxylations here and there, and it will convert the glucose into fructose. I remember this enzyme as PIG. PIG stands for phosphoglucose isomerase. Okay, this step is not very important. It's your wish to remember it or not. So here is fructose, and here is the phosphate attached at 6th position, and this fructose 6 phosphate is again converted into fructose 6 phosphate is converted into a compound called as fructose 1,6 bisphosphate. So what is happening here? As we already told you, fructose is a 6 carbon compound, right? Already as you can see, fructose 6 phosphate was having a phosphate at 6th position. So now what we are doing, we are adding a phosphate at the first position. This is called as fructose 1,6 because phosphates are present at position 1 and 6. So we are calling this compound as fructose 1,6 bisphosphate. You should remember this one, the difference between bi and bis. Bi means, suppose this is a glucose. Bi means if the phosphates are adjacent to each other. Okay, two phosphates are adjacent to each other, those are called as biphosphates. Whereas bis means, suppose this is a glucose molecule, and here is a phosphate molecule at one end, and here is a phosphate molecule at the other end. So this is called as bis. Bis means two phosphates are separated by a long gap. Okay, so here is fructose 1,6 bisphosphate. So can you name the enzyme here? Oh, okay, you can guess it. It is a kinase, because I already told you kinases, they do phosphorylation. So on the first position, phosphate is adding here, so it is a kinase. So where is this kinase coming from? Yes, it is coming from ATP. ATP is converted into ADP, and a phosphate is added to the first position. So what is the enzyme here? It is phospho. A phosphate group is added to fructose. So phosphofructose kinase 1, or you can say PFK1. Okay, so PFK1 is, or phosphofructose kinase 1 is getting this phosphate on the first position. So you will be finding fructose 1,6 bisphosphate. Okay, so once the fructose 1,6 bisphosphate is formed, so this is the structure here, we can break this fructose into two compounds. Okay, we will break the compound as two compounds. One is like this. We are taking 3 carbons and adding a phosphate on the first group, and here is the 3 carbons, and we are adding the phosphate on the 6th position. As you can see, we are breaking the fructose molecule right in the center and making two compounds out of it. So what is the enzyme here? It is aldo or aldolase. So aldolase is an enzyme which will cut this fructose molecule and make it into two pieces right in the middle here. That's why the phosphate is going on the first position, and this 3 carbon with the phosphate at here. Okay. Now this compound here, it is called as dihydroacetophosphate. Okay. And the newly formed molecule here is actually the compound here is called 3 carbons. 3 carbon is also called as triose. Okay. So the newly formed molecule here is glyceraldehyde, and the phosphate is present at third position. We call it as 3-phosphate. Okay. So as you can see, and this dihydroacetone phosphate can be converted into glyceraldehyde 3-phosphate. Okay. So this molecule and this molecule are the same, but the dihydroacetone phosphate is again getting converted into glyceraldehyde 3-phosphate by the enzyme TIP. I call this enzyme as TIP. Okay. Or triose, since 3 carbons, phosphate isomerase. So again this we are forming this glyceraldehyde 3-phosphate, right? So how many glyceraldehyde 3-phosphate are formed from one glucose molecule? So one glucose molecule forms glyceraldehyde 3-phosphate from dihydroacetone phosphate, and another glyceraldehyde 3-phosphate which is directly formed. So from one glucose we are forming 2 glyceraldehyde 3-phosphates. Okay. So I would simply write glyceraldehyde 3-phosphate here. Okay. So from this glyceraldehyde 3-phosphate, so this is the structure of glyceraldehyde 3-phosphate. Okay. We are again converting this glyceraldehyde 3-phosphate into a structure called as 1,3 biphosphoglykyrate. Okay. So 1,3 bisphosphoglycerate is nothing but, as the name suggests, at first position a phosphate is added and third position a phosphate is added. That's why this is called as 1,3 biphosphoglycerate. Okay. So what is happening here? As you can see, during this thing, a phosphate is being added. So can you call this as kinase? As you already know, whenever kinase do the phosphorylation thing, right? No, you are wrong here. Actually, the enzyme acting here is glyceraldehyde 3-phosphate dehydrogenase. Actually, the function of this dehydrogenase is to convert NAD plus into NADH plus H plus. Okay, that is the function of this dehydrogenase. What is happening here? The NAD plus is accepting some protons here, protons or electrons. For the sake of this lecture, I will simply write protons here. That's why you are getting protons added here. So what is the function of this dehydrogenase? They are converting this NAD plus into NADH or plus H plus by adding protons. Okay, protons and electrons are being accepted here. And not only that, so here is NAD plus is getting converted into NADH. A proton is being added, so NADH plus H plus. Okay. Not only that, during this inorganic phosphate is added. Okay, this is an inorganic phosphate, so it is not coming from ADP or ATP, so it is an inorganic phosphate. So that's why a phosphate is also being added here at the first position. So we get the compound 1,3-BiPG. So what is happening to this NADH and H plus? Normally NADH plus H plus are having protons or electrons, right? So this proton and electrons will enter the electron transport chain. I already told you that glucose enters glycolysis and then Krebs cycle and finally enters the electron transport chain. And this NADH are like checks. Okay, they are like checks. So in order to get the money, this NADH goes to the bank, which is the electron transport chain, and this in the electron transport chain, for one NADH plus H plus check, you will get nearly 2.5 ATP. Okay, so you remember that one. And after the 1,3-bisphosphoglycerate is formed, it is again converted into, yes, it is converted into 3-phosphoglycerate. So what is the enzyme here? Yes, it is phosphoglycerate kinase. Okay, so as you can see, you can think why. So just think, a phosphate is getting removed here, right? At first position the phosphate is getting removed. So why are we calling this a kinase? Because of a phosphate is being removed at the first position. So you should know that kinases are adding a phosphate to the substrate. So here what is happening? ADP is the substrate for this kinase. So what it is doing? It is removing this phosphate on the first position and adding this phosphate on the adenosine diphosphate and making it adenosine triphosphate. So here the function of phosphoglycerate kinase is to break the phosphate from the 1,3-BiPG and add that phosphate to the ATP. So it is doing phosphorylation of ATP, not phosphoglycerate. That's why here the function is slightly altered. Okay, so the newly formed compound, since one phosphate is getting removed from the first part of the BPG. So what it is, at third carbon, wait a second, so this is the 3-carbon compound with the phosphate attached at third position, so it is called as 3-phosphoglycerate, right? And again this 3-phosphoglycerate by the enzyme mutase, it is converting into 2-phosphoglycerate. It means the phosphate is shifting from third position to the second position. That's why it is called as 2-phosphoglycerate. And again this 2-phosphoglycerate is getting converted into phosphoenol pyruvate. During this 2-phosphoglycerate, water is removed and Pepsi is formed. Okay, I remember like that. Pepsi is formed, which is hence the enzyme here is Anaerobic. So Anaerobic, it removes the water from 2-phosphoglycerate, and if you remove water, you will form Pepsi. Okay, in order to remember this, I will be speaking like that. So Pepsi is formed, and this Pepsi, okay, Pepsi is a 3-carbon compound with phosphate on it. Wait a second. phosphate is on the here. Okay, so Pepsi or phosphoenol pyruvate, phosphoenol pyruvate is getting converted into pyruvate by the enzyme. Yes, it is pyruvate kinase. So what it is doing here, ADP is a phosphate is being added. As you can see, this phosphate here is being added to the ADP, and ADP is converted into ATP, and finally we will be seeing a 3 carbon compound without any phosphate. This is called as pyruvate. Okay, so this is happening during aerobic glycolysis. Aerobic glycolysis means it is happening in the presence of oxygen, whereas anaerobic glycolysis, anaerobic means there is no oxygen. Okay, if there is no oxygen, as you can see, while this happens during anaerobic glycolysis, this happens during exercise. During exercise, you are doing vigorous exercise, right? During that, there is a lot of decrease in the oxygen. So what will happen? This pyruvate, normally this pyruvate will enter Krebs cycle, and it will form acetyl CoA in the presence of oxygen. This is aerobic glycolysis. But in the absence of oxygen, what will happen? This pyruvate is getting converted into lactate, or we can simply call it as lactic acid. And this acid contains what? Acid contains protons, and protons, what we will do? They will do cell damage, right? That's why we are getting pain when we are doing exercises. Okay, these protons will be damaging the cells there, and we will be getting pain, and we will stop the exercise. So that is the importance of this one. So what is happening? Glucose is getting converted into pyruvate. The end product is pyruvate of the glycolysis. So in glycolysis, one glucose is getting converted into pyruvate. That is aerobic glycolysis. But in the case of anaerobic glycolysis, one glucose is getting converted into lactate or lactic acid. So we will see what is happening here. How many ATPs are produced here? One glycolysis cycle. One glycolysis cycle, how many ATPs are produced? So you will be thinking, how many? First, glucose is converted into glucose 6-phosphate. During this thing, adenosine diphosphate is converted into adenosine triphosphate. Adenosine triphosphate is converted into adenosine diphosphate. So we are stealing one phosphate from ATP, and we are adding this phosphate to the glucose 6 position. So we are investing 1 ATP, or we can say 1 ATP is lost. Okay, first we have to invest, then we will get the returns, right? In real life also. That is the first step. Next step is fructose 6-phosphate is converted into fructose 1,6 bisphosphate. Okay, so we are again converting the ATP into ADP, right? So again one phosphate is lost, and it is added to the first position. So again we are investing 1 ATP here, right? So totally we are investing 2 ATP. Now we will take a look at the returns. So you know that during glyceraldehyde 3-phosphate, by the enzyme glyceraldehyde 3-phosphate dehydrogenase, it is converted into 1,3-BPG, right? And during this, NAD is converted into NAD plus is converted into NADH plus H plus. So NADH, how many ATP it gives when it goes to the electron transport chain? It gives nearly 2.5 ATP, right? So we are investing nearly 2 ATP. Okay, I will add this finally, but you just listen to this one. The second thing was substrate level phosphorylation. I will just explain what is meant by substrate level phosphorylation. Okay,